had the internet, and I started, Googling or Yahooing or also visiting the different drugs she was on, and different things that her medical records said. I was trying to get an idea of what was going on with my mother, and why she couldn't walk anymore, and why she was saying she was in intense pain. And that kind of led me through a bunch of different biofields, including ecology and I got this book. Yeah.
Kickstand.
So I've done some ecology and environmental restoration out in the West, and then I started reading about biofuels, and I became interested in bioengineering and getting cells to pump out oil molecules and just skimming it off the surface of an algae growth pond. And it turns out there's a lot of other people thinking along the same lines. Lots of different methodologies that are being applied. But what's central to the engineering side of things, the bioengineering side, is having DNA. And starting 50 years ago or more, we figured out that we could sometimes take some material and transform it, add it to another bacteria, and that bacteria would gain characteristics of the former. And then we learned that material was nucleic acid and DNA, and we started figuring out, if we can get a certain size fragment, that always works. And those are usually much smaller than the genomic DNA. They're called plasmids. And so these are sort of auxiliary DNA pieces that can be, between certain organisms, be just easily handed off or acquired or picked up and add new functionality or mutate their other inherent functionalities into something new or broken. So at the core of all this is having DNA, and I was reading about all these cool things that I was interested in, and there's a paper, a really cool organism. They found some really cool piece of DNA that did some awesome thing, but they were isolated from the environment and stored in Stockholm and in some old guy's freezer that doesn't answer email. But then a lot of papers in the past maybe 35 years started having DNA sequences of these pieces of DNA, and now we have a digital text file of these pieces of DNA, these parts. So why do I need to cut them out of something I found in the soil? Why can't I just make the DNA from the text file? Have a text to DNA converter. So I've been focusing for the last 5 years on DNA, and more recently, in the past year and a half or 2 years, I've been focusing on detecting DNA and making DNA. And I've come up with a few ideas, and I've done a few experiments, started some projects that are open source. They haven't come to fruition yet, but they're in progress, for sure. So the latest thing that I've been working on is DNA synthesis, and DNA synthesis, because it's really expensive right now, and it's not something that people think that they can do. It's something that the industry and academia thinks that belongs in some huge warehouse, core facility that, nobody makes their own DNA. It's just more efficient to order it from the big factory. But I don't think that's true, because every cell in our body makes DNA by eating, McDonald's, which is $1 for a hamburger. So there's got to be a way to do it, approach biology, cell division, DNA replication, synthesis, price-wise. Because today it's 30 cents a base pair. For 30 cents, I could get, a lot of nucleic acid from a piece of food. So there are some, complications with that mentality, because DNA isn't really synthesized in cells, Denovo. from a text file or from some other molecule, it's replicated, and it's changed or added to by being mutated. So you kind of have a chicken and the egg problem if you start asking, well, where did the first DNA sequence come from? And there's a whole, there's probably a whole textbook or 5 on that topic. But in the beginning, there was a DNA sequence, and it was copied, and every once in a while there'd be an error. One thing would get changed, or one thing would get added or not added, would get skipped, and things break, and then we have something called emergent properties, which, it's like, why do traffic jams happen when there aren't any accidents, and, all the red lights are timed properly at the exit? It just happens. It's just something, feedback in the system of drivers, it's just not all coordinated. So, emergent property of DNA mutation is, if you believe in evolution, us and bananas and monkeys, and we all share like 99% of the same DNA, but we're all very different. So there are some natural ways that approach actual synthesis of a new piece of DNA, and those are, they're generally used for like capping the end of messenger RNA molecules. So when you have a gene and you want to turn it into a protein, it gets transcribed into RNA, and then it gets like hardened a little bit by putting a protein on one end. Maybe it's not a protein, it might be something else. But one end has some hardened modification, so it's not chemically active as much. And the other end has this long string of A, the amino acid, the nucleic acid A, called a poly-A tail. So where does this poly-A tail come from? It's from an enzyme that's similar to the DNA copying enzyme, but it doesn't require a template strand, and it only adds A's. It doesn't add A, G, C, or T. It only adds A. So now I found an enzyme that, okay, it only adds one nucleic acid. It's discriminatory against the rest of them, and it doesn't require a template, which means if I just add this enzyme with some nucleic acid, it's, and maybe a little bit of starter piece, a couple nucleotides long, it's going to make this like N, X number long DNA that's all A's. So maybe there's a way to look at that structure of the molecule and look at the structure of the other replication molecules called polymerases in general and figure out, okay, why does it only add A? Oh, and all these enzyme operations, they're really just really tiny tools. So if the screwdriver bit doesn't fit in the screw, it doesn't turn. So if the enzyme, if the protein has some like extra carbon sticking off in one, volume of space, then another molecule might not be able to fit inside there. I can't wear a 4-finger glove, but a 4-fingered person could. And that's a common analogy. It's like a lock and key mechanism of proteins and small molecules, or proteins and other proteins. So there's also chemical synthesis. So that's sort of experimental, and that's going to require another 10 or 20 years or 30 years of research to figure that out and to make it like a really usable system for making DNA. But if and when it works, it's going to be just as cheap as going and buying any material that has nucleic acid in it and chopping it up into little bits, separating it, and then Anyway, the alternative that is real and existing today is chemical, laboratory synthesis, organic synthesis, and that has there's a cycle of reactions And because DNA is a chain, the cycle is something like take a block, add another block, fuse them together, rinse everything away, bring another block in, fuse them together. And there's activation steps and protection steps so that other reactions, like it can't get added to the back side of the DNA. It can only get added to the front side of the DNA. And the DNA can only extend by one nucleotide at a time. Like if you're stacking Legos on top of each other, the new Lego that you put on and stack every time would have a flat cap that didn't have any little bumps coming out of it. And then you take the cap off, put that cap on your next single Lego, bring it back, stick it on, now you have. And so in the one reaction, when you have all these different single Legos and this one, stack of Legos, because that cap is on there, multiple Legos can't stack at the same time. And until you remove, until you rinse everything away and pull that cap off. So I don't know what everybody's level of understanding is. I don't know. Is there any questions at this point?
So how long does that existing process take to, say, make a plasmid?
I think it's about a minute per nucleotide, or a minute and a half per nucleotide on like the 10-year-old machines, which are comparable in reaction time for the current generation stuff, but the current generation stuff is a lot bigger and a lot more parallelized, and instead of it being the size of a mini fridge, they have machines that are a third of this room size, and they're making not a couple milliliters, but liters of the same DNA strand, or not even necessarily plasmid, because most of the synthesis reactions, they have an inherent error rate because. Do you know what open loop and closed loop feedback is? Does anybody not know what that is? So if you're driving in your car and you're like, okay, I want to go 40 miles an hour, you look at the pedal, you look at the speedometer, when you get to 40, you just stop looking at the speedometer and don't move your foot. And you're like, well, it was at 40, so, even if I'm going up a hill, I'm going to assume it's still at 40, or if I'm going down a hill, I'm still going 40. Closed loop feedback is when you're always looking at the speedometer and you're like, oh man, I need to give it more gas. Oh, I need to take less gas, I need to remove gas. So DNA synthesis is open loop right now. They don't check to see if an addition has been made because they're adding to millions of molecules in the same container, in the same pot. So you can't, they're not working with a single DNA molecule and looking at it, like make sure it grows longer. Oh, it didn't grow longer, we better, repeat that step again. Oh, it still didn't work, let's do it a third time. Oh, okay, there it worked. Or, maybe something happened, and this is very rare in this synthesis, but maybe a double addition happened somehow.
Sure.
Like the cap wasn't properly completely on the incoming nucleotide.
The error rate very high right now?
Well, because it's the error rate is statistical, it's multiplied per reaction. So even though the error rate's like 1% or something, or half of a percent, as you multiply that, the error increases really, really fast. Because half percent times half percent, or if you would want to do the inverse, like 99.95% times 99, or 0.95, and each nucleotide would be another multiplication step.
You mentioned that it would be difficult to create DNA de novo where you're starting from scratch and you want this unique sequence, and so you add one nucleotide at a time. That I can understand that being hard, but if the end result is going to be sort of something that's going to reproduce and similar to the existing species, you don't need to start de novo. You could start pretty much with something that you're close to and then put in your tweaks that you want. So how much easier would it be to begin with a functioning species and run, I don't even know what the sequence would be, sort of like a PCR that would introduce your plasmids to do your dragging and dropping, but you start from like a much larger whole versus Denovo?
It depends how, are your tweaks other pieces of DNA that you already have, and you're just inserting them? If your tweaks are totally de novo, then you still have to come back to DNA synthesis. Sure, maybe you only need 10 base pairs of tweakage, but if they don't exist anywhere, you still have to synthesize those. And it's still expensive today. And the machines that exist are still in some factory that requires, as one of the steps, being mailed to you, or carried in a tube from that machine to another, rather than all being in the same unit and I want to mutate this gene 1,000,000 times. That means I need 1,000,000 different pieces of tweaked DNA. You don't want to have to carry 1,000,000 tubes. You want the output of that machine to go right into the next experiment.
Well, when Venner came up with his 500,000 base pair, did he go de novo, or did he start with something that was?
He started with a text file and broke the text file up into small chunks, and then put that text file into a machine, and what came out of that machine gets put into a plasmid and then amplified in E. coli, and then all the plasmid is extracted from the E. coli, and then PCR sequencing reaction is done, or next gen, and then each of those plasmids are sequenced, and then the right one is kept. So it's closed loop feedback, but it's on the order of maybe 4 days if you're in a really hot shot lab. Maybe, that would take a single person. I mean, if you were DIY or a startup company or something, you've got no chance unless you've got $1 million grant or, a couple $100,000 at least. Because the feedback time is still 1, 2, 3, 4 weeks, just to figure out one stretch of 500 base pairs. Or you can order out, but I mean that isn't what Venner did essentially. I mean they did order out, but that ordering out was still part of the experiment. That if you count the time, if you don't look away from I'm just getting DNA, that's still going on in the background at these factories. They're still doing all this roundabout check work that it's long, but it doesn't. We haven't gotten to the next level yet. So there are some interesting ways that you can synthesize single molecules, and that is instead of having a reaction pot that's this big or even this big, you have it 1 billion times smaller than that, and you have it such that your reaction volume is small enough that maybe only one molecule can really fit in a certain way. So if the DNA is, I think it's 30 nanometers wide, or 20-ish, it's 20 to 30 nanometers wide, the double helix. If your tube that you're putting it through is even a micron, you're already at tube is this wide. It's wider than the whiteboard can be. So if you can make this tube really small, on the order of a couple 100 nanometers or 50 nanometers, then when you push a bunch of DNA from a big pot back here through this tube, you're going to have a greater chance of there only being one molecule in there. And if you dilute this pot back here that you're pushing through full of DNA, you can trap one molecule in there. And then, if you can attach that to the side of the wall, now you have a piece of DNA in there, and you can flow in single nucleotides at a time, and hope that your nucleotide happens to be right in the vicinity where an organic reaction can occur, and it'll couple. Or if you were trying to do this with the enzyme terminal transferase, I sort of mentioned it. Maybe I didn't use that word, but that terminal transferase is a polymerase. It doesn't require a template, but it doesn't just add A's like the poly-A polymerase does. It has A, G, C, or T, but it's indiscriminate. It doesn't have any, maybe certain nucleotides fit a little bit better, so they have, it's not, an even spread, but it's pretty close of which nucleotides it'll add. So if you had an enzyme floating in here in, like, say, a nanopore sieve, these exist that have tunable pore sizes. If you had a sieve back here, like a filter, then you could have your DNA, which is big enough to get through the filter, but it's attached here on the wall. And then you can have your enzyme, which is going to be really big, and then, you know, you flow a single nucleotide in here, and this would be the terminal transferase, and you would still have to wait for diffusion to happen, because if you're only adding one molecule and the chamber's this big, it has to diffuse all over before it hits right the sweet spot where a reaction can occur. And if you know, your enzyme can't be attached because this DNA molecule is extending, so your enzyme is going to be diffusing, your nucleotide is going to be diffusing. This is going to be, the end of this is going to be flapping around. So you've got like three somewhat, not random, but chaotic. You know, so seemingly random that it's going to be really low probability of them happening without enough time just sitting there reacting. Even though the reaction itself, you know, happens in nanoseconds. Diffusion takes a really long time. So, and there are single molecule detection techniques where, like, you can point a laser at this, and it's like a gold or a silver bottom, and you point a laser at it, and you look at the beam that bounces off, and then you put that through a spectrometer, and which sorts the light frequencies spatially. Like if you look at like a rainbow, that's white light being broken up by water droplets this way, out. And use that's called surface enhanced Raman spectroscopy. But then you need a gold bottom, and the DNA molecule has to be there. So it's too much of a hassle that won't work very easily unless we have really precise nanofabrication techniques, which we can't really build single atom at a time right now. Intel is doing, like, I think 22 nanometer or 15 nanometer transistor processes right now, which is pretty small, but there's still margins of error in that. So, but that's still pretty good. But you know, that's transistors. It's not like a tube with an empty volume going through it. So at the last talk get-together that I stood up in front of and talked about this stuff about a month and a half ago, some people mentioned, well, hey, why can't we, you know, what if you could do a bulk reaction where you still got a billion DNA molecules, and you add a nucleotide, and you're not sure that a nucleotide has actually been added, but, you know, there's this thing called gel electrophoresis that sorts DNA molecules by size. So what if you did a DNA reaction, you added a single nucleotide, separated the DNA by size, cut that chunk out that's, you know, n+1 of your DNA molecule, cut that chunk out, and put that DNA into your next reaction. Now you're filtering out your error at every addition step. And that is actually something I haven't seen, probably because it's traditionally gel electrophoresis is this big, and DNA synthesis is mailed to you, so people like haven't really tried this, or people that do have a DNA synthesizer, they put out, you know, a vial, they spit out a vial of stuff. It's not like people just haven't done that before that I've seen. So that is really cool to me.
What do you start with in this?
In this
situation? Yeah. Do you just order whatever board, or?
No, you would get like this could be like an iron bead that's coated in plastic, and then the outside of the plastic coating is modified with carboxyl groups, like COOH. And so this is on the plastic bead, and these are reactive with pretty easy to use kits that you can add nucleic acid or protein to this right here. A couple simple steps. So you would start with a bead like this, and it has an iron core. And so now you can put a magnet up here, holds your bead, and with this right here, you would just, instead of adding DNA and doing the fixative step to attach a long string of DNA, you could just add like a single nucleotide. So then you'd have, I don't know where it gets added to exactly, this might come off, but then you'd have, you know, an A to start with, attached here or something. And then you put this in here, and you do your synthesis reaction.
When you add one nucleotide, what's to stop it from adding multiple times and adding a bunch of A's in sequence?
So, nucleotide without a base is pentose sugar, which means it has 5 carbons. So there's 1, 2, 3, 4, and then there's a 5th carbon here. And the base, the A, G, C, or T, attaches up here. And I think it's numbered by the IUPAC system, which it's a naming scheme for molecules. Instead of calling this a nucleic acid, it's, you know, 1, 3, 5 ribulose. I don't know what it is, but it's some, you know, really long, and it's machine readable. Let's say that. So this is like the most electronegative atom, and that's generally where numbering starts from. So this would be the 1' carbon, this would be the 2', 3', 4', and 5'. And if you know anything about DNA, you'll have heard, you know, oh, the polymerase goes from 5' to 3'. You know, it copies 5' to 3', and that's talking about which way the molecules are getting linked together. So up here, attached to this 5' carbon, there are a couple of phosphoryl phosphate groups, which is PO4, and this, like that, and then there's the same thing repeated, another. This thing again. So these 3 phosphates they have a lot of energy in the bond, and when another nucleotide comes in, it has, you know, this 3' end reacts with these, and the energy is enough from breaking this bond to attach the 5' to the other 3' through this oxygen. Or maybe it's on this oxygen. Yeah, it's on this oxygen. It has a phosphodiester backbone. So you would have here an O, P, O, O, and then you'd have your 5' of your, this is the 5' of the other nucleotide. And it's the phosphodiester bonds. These are ester linkages and diester phospho. There's electrons. I think that's the right number. It might be that many, but there's some negative charge here because there's some electrons that aren't bonded in those oxygen. So DNA has a negative charge generally, overall, and that's how electrophoresis works. You put this molecule in a gel, you put the negative terminal of the battery here, put the positive terminal here, and the DNA, negative, positive, opposites attract. DNA goes that way in gel electrophoresis. I don't know why I was talking about that, but
How do you keep it from double
How do you keep them double attaching? So in a synthetic reaction, they don't use the ATP over here, or it was an ATP, but doesn't use the triphosphate for the energy. It uses something called there's a couple different ways to do it, but basically it uses a different chemistry, and it doesn't go in 5' to 3', it goes in the opposite direction. So there is basically, so if you have, this is your starting, this is your magnetic feed down here, we'll say, or whatever, and you don't have this other nucleotide here yet. This 5' side is reactive. It's attached to a solid support, so it's not moving around. It's reactive, and then you flush in nucleotides. But the nucleotides that you flush in, the 3' right here has been made, or rather, so the 3' is activated so that it links up with the 5', but the 5' up here has been deactivated. It has a protecting group on it. And once the linkage is made between the two of them, all the free nucleotides are flushed out, and then a decapper or deprotectant is brought in, which takes that X away and makes it good to go for the next reaction. And you flush in new nucleotides. That are capped, reactive at the 3'. It links up. This is no longer reactive. All the free nucleotides are flushed out. Deprotectant comes in, deprotects that. New nucleotides are brought in, and the cycle continues. So,
Pretty efficient.
What?
Seem pretty efficient.
Except you have the diffusion to worry about, and this, I'm showing you a blown up image of making a single molecule, but in reality there are like billions of molecules being made, or tens, hundreds of billions. So since they're all floating around in this reaction chamber, and diffusion is what's limiting your efficiency, if one nucleotide, if 1% or half of a percent of the billion molecules don't get the addition happening, then on the next synthetic reaction, on the next cycle, 0.5% of your molecules are garbage already. But they don't check every cycle. So by the end of 50 cycles, you've got a whole clan of similar but different molecules. And a large portion of those are good, but they're all mixed in, and then you've got to separate them. And separation time is, 4-day to 4-week process, depending on how.
So you propose run it through microfluidic and then do a check on every single reaction?
Yeah, not nanometer scale, just micron scale microfluidics. I think 10 by 10 by 40 microns.
Well, before we get into the microfluidics approach, the way nature is adding a nucleotide at a time is this template happening. Well, sort of, you could use the ribosome as the engine that's doing the addition. Have people Well, ribosome is making proteins, but it's also, could be tweaked to be the tool that adds the next ATCG onto your DNA.
I would think that you'd better start with the polymerase, because the ribosome is optimized for catalyzing amino acid linkages, not nucleic acid. They're different structures.
Well, functionally ribosome, but for the purpose of, for us, we want to add nucleic acid now.
Yeah, so I would start with, we already have a crystal structure of a terminal transferase, dideoxy terminal transferase. We already have a crystal structure of that and like a host of other polymerases. So these are all adding nucleotides. We have knowledge of photoisomerization linkages. So like if you have like a, I don't remember what a ketone looks like right now, but ketone, enol, isomerization, it's a certain linkage where you have a carbon here and then like, I think it's maybe another oxygen and something. I don't exactly know, but basically this double bond shifts around and that's called isomerization. So it goes from having a hydrogen here and then, that disappears and the hydrogen pops up there, and it's certain, certain things induce this shift, or it's just random with heat. But they found photoisomerizational groups where if you pulse it with a beam of light, then that double bond swaps over there, and because electrons are moving around, it changes the whole electron 3D space, which changes the conformation of the protein. So if the double bond here, double bonds are rigid, they can't rotate. So if you pulse it with blue light and all of a sudden it can rotate, maybe it was bound up and it couldn't add a G because it was kinked the wrong way.
Right.
Pulse it with light, now it's free to rotate, now it can add a G.
Well, how about this?
And not add ACT.
Well, what if you, okay, the functional equivalent of ribosome that has the ability to add additional chains over time. But the problem is its current function is based off the template where it's just doing a 1-for-1,
3-for-1.
right, something 3-to-1, 3-to-1, 3-to-1. Well, what if you have the functional aspects of ribosome that allows you to add additional chains, but you use a trick similar to what you just mentioned, where you pulse it with something that forces it to do a hard conformation, like some bending thing, which will only let it add chains of a certain A's, AA. And then another pulse to tweak it so that it's only doing G, 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 G. And then, so that, you've got a desire of a sequence that you want, but that keys off the pulses that you're giving it, so that this mechanism is creating a whole sequence of chains, because it'll be too fidgety to have one chain per pulse. But what if you tweak it, pulse it, so that it's making a whole series of the same chain, and then you pulse it, and then it goes to the next in your sequence. And then you have your Denovo sequence, except it's a whole duplicate. Instead of ATCG, which is your desired sequence, you have AA, TGG, GGG. And then the next step would be put it through a similar trick where you're feeding in this long chain that has the dupes, but every time it does a toggle to the new flavor, it pops out so that after the second iteration, the duplicates are just triggering singles so that you got your single sequence.
I think you can do single nucleotides at a time, and don't have to go through the whole duplicate thing like you're saying. Like the crystal structure of the terminal transferase, it sort of looks like a baseball mitt, and I guess like this, as I remember it. And the DNA is going through this way, or the DNA is like right there, it's being held, and then a free nucleotide comes in, and this finger sort of pops it in to the reaction site, and sort of all at once in that popping action, it condenses the molecule, the nucleotide, to the rest of the molecule. And then sort of like a semi-automatic pistol, or actually an automatic pistol, it pops it in, it fires when it condenses, releases some energy, and then pushes and ratchets it this way. So now you have another nucleotide there, and the finger opens back up, and it's ready to accept another nucleotide, pop, ratchet. And there's actually been some experiments or some research that they showed. I believe they had two plates, and they attached a polymerase over here and like a starting RNA, or maybe it was a ribosome, and they were making protein. But basically, they constrained the spacing on these plates. So whatever it was, I don't know if it was a polymerase making an RNA molecule from DNA or a ribosome making protein from RNA, but whatever it was over here, it has that ratcheting, both polymerase and ribosome have that ratcheting action. So what they did was they had each molecule tied, or a protein's a molecule, but they had the two sides of this reaction tied on these plates, and the plates were on screws. And so they would add the stuff, the protein, the amino acids or nucleic acids, and it would add one, but then it couldn't ratchet because, well, I guess if it's making it this way, it would push it up. So it was I don't know, it was one of these ways. But basically, if it's pulling it through this way and making a new strand here, the old strand's going this way, so it needs to pull this way to get the next frame in to read or to replicate. So they would add stuff. They would add a monomer to the polymer, but then because it was affixed on these plates, it couldn't ratchet. And then they would turn the screw and give it a little more space, and they would see exactly how many nanometers of screw turn caused until the molecule got one monomer bigger, and they were actually able to see. They may have also had like a tension meter over here to see what the force that the enzyme was pulling on the molecule, like the ratcheting force. So they effectively, just by tugging on the molecule while feeding through, they made it from zipping through to one at a time. So if instead of pulling that, you just whatever find a key linkage in the protein that's twisting around when it's ratcheting, put a photoisomerase, blue light photoisomerizable, unit on bond there, and only when you pulse it with blue light, now does it able to relax and ratchet through. And now you've got your stepwise. Okay, go ahead. Next one. And like I was saying, that the terminal transferase sort of has like this finger action, where you have your molecule, you have a new nucleotide, it pops on and then it ratchets and it opens back up. It really is it sort of looked like a finger to me on a hand, where it had this sort of bending action. So maybe you put a photoisomer, I don't know how to pronounce that, but maybe you put that photo-conforming group on my knuckle here, and then only when I pulse it is it able to pop a new one on. And then maybe instead of having just one thumb that can accept all 4 nucleotides, I just add 4 thumbs onto my hand, and each one of those has a different color isomerization group. So now I can have a whole mix of nucleotides, A, T, C, and G, all in the same pot, but only when I pulse with red, green, blue, yellow does it, one of my 4 fingers goes, okay, A, T, G, C, A, T, etc.
But they have the molecules that are sensitive to 4 different kinds of pulses?
I'm not sure. There probably is at least a couple. I don't know if there's 4 or 5. But I imagine they do, or it's the mechanics of it is relatively figured out where it's achievable if you wanted to apply. If you wanted to do that, you could get to it with the right team and an achievable short period of time. Whether that's a month or 2, 3, 4, 5 years, I don't know. But I think that stuff is relatively. I don't know. I don't know if it's well figured out or not, but I think it probably is. So that would be awesome. A controllable polymerase, a digitally controllable polymerase would be great. That would, then you could really just take a piece of food do a DNA extraction on it, add nucleases, chop up the DNA, purify all the nucleic acid out of the sample, activate it with the ATP, and flush it into your enzyme chamber, and just go pulse your light. Then your reagents are essentially just coming from like Mr. Fusion and Back to the Future. Whatever it is, dump it in, get the DNA out. So what I've been doing for the past 9 months or so is, I've been reading about and learning about microfluidics for probably about 3 years now, and I took a lab course at the school I was going to in microelectronic fabrication. So I made some transistors on a piece of bare silicon, and I got sort of an idea of some of the micro-scale fabrication techniques, and nobody was really doing microfluidics at my school, but I tried to pursue it anyway, and I ended up using a Versalaser, Universal Systems Versalaser desktop CNC laser cutter, CO2, like 10.6 micron frequency beam, or wavelength. And I have a test pattern, a microfluidic test pattern with, I think I made it in Inkscape, and it's FDG, and basically all that is a bunch of different line thicknesses, different number of pixels in Inkscape, this way, this thick, this thick, this thick, and then I think I did some curves, like a corner or something like that, and the Air Force has a similar test pattern for I think visual systems. So take optical replication, how well do all the lines reproduce when you keep taking pictures of the photo that comes out, and so I printed a pattern like this on this silicone called PDMS, which it's basically like RTD that you buy at an auto parts store, room temperature vulcanization silicone, which means that silicone oil exists, and vulcanization is when you cross-link the silicone molecules, and it creates like an elastic structure, an elastic matrix structure. And I think vulcanization was discovered by Goodyear, maybe? Whoever was the first guy that made really good tires. But so I printed this pattern with just a off-the-shelf laser cutter, and I looked at it under a microscope. I looked at it under an interferometer, which an interferometer uses a laser that is a really good laser, and the frequency of light is very consistent, and the beam is really straight. So if you shoot a laser beam at a surface and it bounces back, it's going to there's a thing called interference, and it can either interfere constructively or destructively with itself, and basically if you have a laser coming this way, it's going to bounce off and go this way, and if you had a beam splitter right here, like a half-silver mirror, half the beam would go through this way, half the beam would bounce this way. But this difference right here, the difference in distance between this reflection surface and this reflection surface, if it's not an integral number of the wavelength, if you then combine these beams back together, if you put a mirror here, a mirror here, or actually rather, have a prism there, and they come out here. If this isn't an integral number of wavelengths distance, the beams would, the waves, like, you just cancel each other out, and you wouldn't have any light coming out of here. Whereas if it was off by half of a wave period, when they combine together, the phase of the light beam would match up, and they would constructively interfere. But since you just split it in half, it's not really giving you. You're not making light out of nowhere. But if they were, say, like I said, off by if they were completely out of phase, they would totally cancel each other out. So if there was a dip here and a peak there, you know, when you it's not multiplication, but it's
Just adding them, right?
It's just
adding.
Cancellation.
It's just cancel now.
Yeah.
it's not. Okay. So yeah. So you know, and the output of this would neither be. It wouldn't be a squished wave, which is
Almost the same magic is off of the space. So that's why.
like where is it? I'm trying to.
Phase cancellation.
Yeah, it's a phase
Sure.
cancellation. It's easy to understand.
So because of this property, if you had a sensor, a light sensor here, you can tell the distance between these two plates depending on how bright it is down here. Because you know the brightness of your original beam, and depending on how much interference is or isn't happening, you can tell, oh, they're perfectly one period away from each other, or they're half of a period away from each other because there's half as much light, or if they're totally opposite phases, you'll get no light or very little light out here. So if you move this plate, the amount of light your sensor would change. And instead of having a plate here, if I put my 3D channel made of silicone and I move it back and forth, now it's reflecting down here, and I'm moving it, it's reflecting down here, and then all of a sudden it's reflecting up here when I'm scanning it through. And if I stitch together my sensor readings, I can get a 3D profile.
And
All I was trying to explain.
so what? So moving that.
They don't actually move it, they just use a camera chip. So it's got a bunch of pixels instead of sliding it.
So then through software you could recreate the geometry of what the real surface is.
Yep.
Mm-hmm.
You do it.
And you can make an interferometer really easily with like a CD and a CD case and a handheld laser pointer.
What is this called?
A Michelson interferometer. If you Google $20 Michelson interferometer, you'll see a little video on some not YouTube video site. And it shows this like super glue, some gum bands or something, and a CD and a CD case, and he's got it shown on his wall. You can see the interference pattern, and it looks like it's called a moire pattern, and it's bands of light. And as he bumps the table, the light bands move back and forth. And they move back and forth because that sensor is only at one, if your sensor is just in the center here, when these move, this moving back and forth is actually variations in brightness. Dark light, dark light, dark light. And that moving pattern on the wall when the guy bumps the table, it's basically showing it's moving, a certain number of nanometers. There's nanometer scale vibration on that optical system. And they use that to do three-dimensional profiling. So I did this. I made these microfluidic channels with a desktop CNC laser cutter, and the 3D profile showed that they were junk. The walls looked sort of like the edge of a block of Swiss cheese. And I attributed that to a number of things. It was a desktop laser cutter made for cutting at the smallest, like balsa wood. Maybe you could cut a 250 micron line, like thick section could be cut out. So that's a quarter of a millimeter. So that's fine for model rocketry, whatever, remote control airplanes, putting together your MakerBot or your OpenPCR wooden case with all the tongue and groove stuff. But 250 microns is really big for microfluidics. It's right on the upper edge. And the walls look like Swiss cheese, which I attributed to because it's made for millimeter scale stuff, not micron scale stuff. The stepper motors aren't doing small enough steps. The laser is pulsing. It's not continuous wave, even though a CO2 laser can be run in a continuous wave fashion, which means there's always photons coming out, and it's going to be, if you don't turn the laser off, it's going to be an endless stream of photon wave stuff. So, but a lot of them, instead of controlling the current going to the lasing chamber, they just pulse width modulate the full power. So instead of getting a nice wave like that, or if I just turn the current down, you know, a less loud wave, it's actually full power, but then it's like, so if I'm shooting light that has enough energy, these sections of light have enough energy to burn my stuff, but they're pulsed, and then I've got a stepper motor, which is a if these are out of phase, it's not going to produce a totally smooth channel. So, and you know, that machine was my school's, and I couldn't mess with it. I couldn't tweak it. And the people, it was like in the model airplane club room, and they were like, "Oh, you can't put that, you know, reflective stuff in there. It's going to go back into the lasing chamber. It could damage the optics." And I'm like, well, the laser works by having photons in there bouncing around. Like, it's not, back reflection is probably not going to hurt it, but these guys weren't chemists. They were mechanics.
Have you tried spin casting the DPMS directly onto a like a DVD disc?
I didn't know. I used silicone wafer.
Yeah, if you have like a Blu-ray player, then you would get the fine resolution you need.
I thought of that using the tracks.
Because then also the optics.
But then you have to
increase the power on the dial.
Then you have to convert your SVG to polar coordinates, right?
Mm-hmm.
Well, because I don't want pulsed action, if I'm trying to make a square on a rotating DVD in polar coordinates, as my CD is spinning in this direction, my laser is only, you know, it's only right here. Actually, well, if I'm trying to make a square here, but my laser head can only move in this direction, then as it's spinning, when it comes around, I got to pulse, move the laser, pulse, you know, which I don't want to do. I want it to be continuous wave, and
but I think the problem you're having is the laser, the optics on the laser is not that good. So the spot size is somewhere around 5 mil. And
but the spot size doesn't.
The spot size on something like a Blu-ray player, the optics is a lot finer. It's a lot, so you'll get a lot tighter beam.
Yeah, it's a 400 nanometer wave, 405 nanometer wave.
But the optics itself, it's short focal.
The track spacing on a Blu-ray, I think, is 600 nanometers.
High enough for you to get diffraction grading off of it.
Yeah. It's like 600 nanometer track spacing. But I mean, there's also error correction in reading a DVD. So you know, it's pretty big. It's like, I think it's, I don't know what it is, but I think it's like 1 byte to 20 bytes or something worth of error correction. So that means that even though you're right, the DVD has a spiral on it, and I can convert my square into polar coordinates and pulse it, that head, even though it might be averaging reading that track, it still has error correction built in, so that if it overshoots the track, it's got enough time where it can react with that voice coil and move the head back to the track.
Well, what if you're Okay, so the challenge is that the physical size of the width of the helix coil DNA is too narrow to be able to get down to that resolution with your toys. What if you generate this sequence that you want of the ATCG, ultimately that's going to wind up in DNA, but through your toys, you apply your technique to a much bigger molecule where you do your pulse code and your tunnels and your ion gripper onto a big fatter molecule that's more accessible, but still able to persist your sequence that you want. And then once you get it into this bigger molecule, and then do some magic to walk through that and take that sequence that you've got on the bigger molecule and somehow take it down, you know, chemically to
like Richard Feynman, like just having your machine make smaller and smaller arms.
Exactly.
Maybe, but I think I mentioned earlier that you could have a bulk reaction, add one nucleotide, and then do an electrophoresis and pull off the right size. And if it's the right size, you know that extension has occurred. So that's a possible way to have closed loop for every synthesis cycle. So picoliter DNA synthesis has been accomplished, and that means that the volume of the reaction chamber is on a picoliter scale. And I think 10 microns by 10 microns by 40 microns. I think that those dimensions box is like 7 or 10 picoliters. So 10 by 10 by 40 is basically what I'm trying to achieve with I've been designing and working with a couple people on the internet to build a custom laser cutter essentially. Just using, just downscaling everything, using a finer thread screw, maybe using, for a while I was looking at using like a 400 step per revolution stepper motor, but stepper motors have some issues and, resonation and...
Nathan, it's approaching 8 o'clock. I just wanted people to be, if they had more questions, it's clearly this is generating some questions and ideas. 2 things. What's next for you in terms of this, and how can people contact you? Maybe that.
I'm moving to Portland, Oregon in the next 3 weeks. That's my next thing. My next thing is putting all my stuff in a box.
Yeah.
But I just ordered a Blu-ray laser, a laser diode from a 12x Blu-ray DVD writer, Blu-ray writer. It's like 405 nanometer wavelength lens laser. And I'm gonna measure the angle of the beam and then put that into some open source optical ray tracing simulation software.
And where can people find all this information or about you or contact you? DIY. What does that say?
DIYHPL.us/ I don't know, it's always in my phone. If you Google this, site :DOH plus, and then just type laser _ etcher, if you know how to Google that well, you'll find it.
All of your views. And what's the next, what is your vision ultimately? What would you like to do?
So I'd like to have some, if not laser cutter, there are other ways to make micro stuff at really fine scale. There's a company that for not too much money will print a photo negative at 10,000 dpi using a laser, and they can do really, 10,000 dpi is 25,000 divided by 10,000 with a 2.5 microns. Is that right? Yeah. So if I can't get a laser cutter to work the way I want it to at a fine scale, so that I can iteratively and rapidly prototype microfluidics, because I don't have the time to take 2-3 years